What's up, Reef Builders? Welcome back to another episode of the Reef Therapy Podcast. This is episode 128, brought to you by Cali Kid Corals, the ultimate coral experience. Today, I am joined by Eli Meyer and Andrew Bauma from Aquabiomics and Reefability, respectively. How are you guys doing today? Thanks for coming on.
Pretty good. How about you?
Doing good. Thanks, Salem.
Doing good. So I've heard there's some new things coming from both of your companies. I think skip the introduction. Let's just get into it. What's new from Aquabiomics? What do we have to look forward to in the coming months, Eli?
Yeah. So right around the corner here, next month, starting in April, we're going to be running three batches every month. So that's a batch every 10 days. So big improvement in turnaround time. That's always been the number one pain point for our clients, is waiting for that sample to come in. So yeah, your results could come in as quickly as 10 days now, starting in April.
Yeah, that's great. I remember when I first started using your service, maybe late 2023, early 2024, was one batch a month. So it's a pretty big improvement, up to three now. So great.
Yeah, and it's all about increased sales. You know, the more people that use the service, the more batches we need to run. So keep those samples coming in, and one day we'll get up to 4 batches a month. You know.
Yeah, so something else I heard people really wanting is the rock. So we touched briefly in December that you're now a rock farmer. How's that coming along?
Right. It's been slow. It's been slow breaking down the coral shed, you know. So I set up this whole coral shed, and I was selling corals for 5 years there. I've got 1 tank now ready to convert over to rock, but yeah, it's a slow process. In the meantime, I do have rock and sand at a smaller scale going in the shed all the time in my other tanks. So every month I buy new sterile rocks and start a new batch of the aquacultured live reef rubble.
Nice. Something
still several months away before that's going to be ready to sell.
Slow to grow rocks, man. That makes sense. Yeah,
sure is. Yep.
So something that I really wanted to talk about on this episode that we had spoken about was the lineage testing. So you had mentioned some updates on that. Why don't you share that with us?
Yeah, so this is a project that Andy and I have been hatching for a while now, trying to recruit other coral farmers to join us. And finally we said, look, let's just do it ourselves. So here's what it comes down to. You know, if you buy a coral with a name attached to it, you, the customer, are assuming that means it's really a clone. It's really a piece broken off of that mother colony, and it's genetically identical. That's what the name implies. But is it the truth, or are people just slapping that name on something because it looks similar? I think a lot of us suspect it's some of the latter, right? There's some imposter corals out there in the hobby. And it's an interesting puzzle because it's a question that DNA sequencing can answer. You know, we can have a definitive answer to this question. So here's what Andy and I are going to do. He's collected lots of corals for this project, and Andy, maybe you can tell him, the details about them. But it comes down to this Oregon blue tort, comparing the real thing with other samples that may be the real thing or maybe imposters. So Andy, maybe you can go into some of the samples, some of the history that you have for these.
Yeah, so we've talked about doing a variety of different corals, and we were having a hard time finding a good coral that we had really solid lineage for back to the original import. So we were going to do Walt Disney, but the person who was going to supply us with known lineage Walt Disney lost his colony. And so anybody listening, if you have Walt Disney and you got it directly from Mike Biggar when he was still living, contact us, because we would love to use a small frag. All we need is a polyp-sized sample of your coral for this study that we're doing. So we decided to shift because we're in the Pacific Northwest. The Pacific Northwest is, not surprisingly, where the Oregon blue tort was first imported. And it comes from a shop in Portland called Upscales Fish and Reptiles. It's a shop that's been around for 30 years. It's got a great place to go for SPS. And the owner, Travis, loves blue corals. And so he's got all sorts of blue torts. And I have more than 100 different acro varieties myself, and I also like blue corals. And so I've gotten a number of blue corals from Travis. And so I have a known lineage Oregon blue tort from Travis, because Travis was one of the originators of that piece. There's kind of a long story that we could get into, but I won't. But Travis has solid lineage Oregon blue tort, and there's about 5 other blue torts that look a little bit like it, but are different, say in growth pattern, maybe a little bit different in coloration, and some that he knows came from other places. But they all look relatively similar. They're blue, beautiful corals, and share some characteristics with blue tort. We also have Jake Adams' Immortal Tort, which my understanding is that Jake collected himself. It looks really similar to Cali Tort, which is one that I don't actually have, but is another one is often kept by people that like blue corals. Jake's Immortal Tort is similar to Cali Tort, and it gets really, really bright blue and really high par. And so in my tank, it looks almost exactly like Oregon Tort. So I have that one. I have about 6 different blue torts. I have, I have the original Oregon blue tort from Travis at Upscales. And then I have some that, because of what we know about acrophylogeny, that is the evolutionary relationships between different acros, we have some that are relatively closely related, but a little bit further apart from that group of blue acros. And so Eli has a number of genes that coral researchers use to determine species by DNA testing, and we're going to determine the species and the variation among all those pieces. So we're going to be able to see, okay, this is what the genetic profile for these genes of an original lineage Oregon blue tort looks like. We're going to be able to see, how different is that from these other blue torts that look similar? How different is that from some of these other closely related acros? And it's going to be really cool because this hasn't really been done for hobby corals yet. And it's going to sort of establish, what is possible to do in terms of tracing lineage of hobby corals, so that Eli can say in the future, hey, I can genotype your coral, and I can determine if you can provide me a known lineage coral, I can determine if other coral frags are from that same lineage or not. And Eli, I'll let you pick it up from there. I talked a lot there for a while, so
Yeah,
you pick.
Yeah, exactly right. So, I mean, this project we're doing here is kind of a model for the application we have in mind in the future. I don't know right now, if there were a lot of hobbyists concerned about imposter Oregon blue torts, right? But it's a nice system because we have a good lineage sample and we have other putative Oregon blue torts.
I don't know, Eli. Some people are awfully picky about that. They want to know that their Oregon blue tort is real Oregon blue tort. Yeah. And I have been accused, because I have had relatively good success at growing Oregon blue tort rather quickly. I have been accused of maybe not having real Oregon blue tort because mine grows too quickly. Because one of the characteristics of Oregon blue tort is that it's a slow grower, right? It just sits in your tank and kind of hibernates for a year before it does anything. So this study will help out people like me who people doubt have solid lineage. So yeah, sorry, go ahead.
Yeah. And I'll say, ever since I've been in the hobby, it's something that's kind of irritated me. The probably going to piss off some listeners here. Sorry. The concept of lineage. I mean, somebody says, "Oh yeah, I have this lineage piece." Well, let's poke at that concept. What are they really saying? They have some handwritten notes in a notebook somewhere. I mean, do they even have that? Maybe they have a receipt. Maybe. Very often, it's like it's just their word, right? They're just saying, "No, I got it." well, how is that really different than just putting a name on it in the first place? I mean, in either case, we're just being asked to believe the person. It's not verifiable, right? So I really like the idea of having something verifiable where you can come in and say, no, that is or that isn't, what you're claiming it is.
Yeah, there's a lot of people that just say, "Trust me, bro. It's
Right.
from this
Yeah,
guy."
exactly.
And
Yeah.
I even, like, one of the shops I used to work at, like, torches were very big when Indonesia was shut down in like 2019, 2020. And the Lineage torches were selling for $1,000, $1,500 a polyp. I mean, like, a lot of money was exchanging hands. And we
Yeah.
had some wild stuff when Indonesia first opened up. It looked very similar to some other named, in quotation, Lineage pieces. And a guy came down and said, "Oh, I'll cut you a deal. Let's do like $3,000 cash and I'll buy all of these." We're like, okay, sounds great for us. And then the next day he went and posted them as like TCK Master, TSA Holy Grail, like, because they were
Right.
wide look-alike. So to anyone that doubts that this does happen in the hobby, I think it does. And I think Reef to Reef and your local Facebook groups are very ripe for it. A lot of people are in this to make money, sadly, and don't really respect the word of mouth lineage. So yeah, having a verifiable system excites me quite a bit because I think it can it basically it establishes a pedigree for the coral at that point. You can have a pedigreed coral.
Absolutely. Yeah.
So I guess my first question is, so you're predicting at this point, or your hypothesis is that a lineage or an aquaculture strain of Oregon blue tort will have a genotypic difference compared to, let's say, the original place the very first Oregon blue tort was collected. If someone goes out and actually takes a piece of the colony there and ships it in, you think the aquaculture piece will have a genetic difference?
Yeah. And I think you're right. It's very fair to say it's a hypothesis at this point because we know those sequences. We know the sequences of these genes for these species that we're talking about, but we don't know it for these individual specimens, right? But here's what I can say: the genes that we're targeting are genes that they have. Now, I have to go into jargon here a little bit, but I'll try to unpack it. If you look at the sequence of a gene, which is just the string of letters laid out like a word, parts of that gene are the same in every member of that genus, or maybe even in the same in every member of that kingdom. Does that make sense? There's parts of the gene, we call them conserved. That region of the gene is sort of always the same. And so looking at that part of the gene isn't going to be useful for telling you what species am I looking at, because it's the same in all of them. In contrast, there's other regions that are hyper variable. They're super different. And looking at those parts of the gene, really every different individual may be different in those parts of the gene. Certainly different species have characteristic differences. So what we're doing, we're targeting genes that have known really variable regions, and they're variable regions that are informative for species identification. So one thing that'll fall out right away, and this isn't a hypothesis, we know this already from papers, that right away we'll get a solid species ID on these specimens. Right now people are IDing these things morphologically, which is done widespread in coral research too. You kind of have to when you're out there diving, right?
Eli, I got to
yeah.
stop you for a second and pose a sort of an existential question here. What if your analysis says that the Oregon blue tort is not actually a tort?
Maybe wouldn't surprise me. I mean, coral taxonomies are getting revised.
Now that might make some people mad. I don't know. We have to change the name. The Oregon blue, whatever.
That's a good article. Oregon blue tort turns out not to be a tort.
Yeah, it wouldn't surprise me. Yes, we will learn that answer for sure. One way or another, these data will for sure tell us, yes, this clearly falls into this species, or no, it's a member of a different species. Yeah. But what's less understood is how much variation between individuals is there in these regions. And that's where Andy's collection of corals is going to be really valuable here, because we have a bunch of specimens that we're pretty sure are the same species. And so now we can look at which parts of the gene vary among individuals within a species. Ultimately, it's going to come down to looking at just a few base pairs. We're going to have a few parts of the gene that tell us that is the Oregon Blue Tort. And if you don't have those few base pairs, you're not, you're something else.
So what I'm kind of picking up is that you speculate that because the Oregon blue tort has been in captivity for X amount of years, there's been, some mutations that have occurred that have been relatively conserved at that point through aquaculture, through that artificial selection. I suppose my question would be, let's say, there was the original Oregon blue tort, and let's say like F2, if you would, like the first frags from that colony were made. And one guy in Orlando and one person in California both grew it in different tank parameters. Do you think that if we were to test both of those Oregon blue torts, that maybe there's the traditional track of lineage, they can both be tracked back to that original shop through receipts? What if they come back with some variability between even those? So the process of aquaculture in different environmental conditions within someone's tank shifted them. I mean, how would you compensate for that to still say these are both an Oregon blue tort?
Right. I think it's fairly unlikely for a few reasons. So if we were to sequence the entire genome from those two specimens, then yeah, I think we'd probably uncover some genetic differences. But only if we were to sequence it from sort of all parts of the colony. So think about this. When a mutation emerges in that frag, it doesn't emerge in all the cells at once. It emerges in one of those cells. And then that would have to basically grow out to a whole branch. And somebody would have to break that off in order for us to detect that as, okay, this is a new characteristic mutation. And I believe that will happen at some really low level if you're talking about the entire genome, right? But we're not. We're talking about just a short stretch of just a few hundred letters. And what are the chances that that happened in that few hundred letters that we're looking at, and it's swept to completion, right? So that this entire new colony has that thing. So I can't say it's impossible, but boy, I'd be shocked if we see that. And let's do it. Let's test a couple. Certainly we should do duplicate frags from a single colony, just to confirm that yes, you get the same answer if you genotype the same colony twice. But we could also do frags that were generated from a colony and have some lineage kind of relationship there. I don't know, Andy, do you think you have anything like that?
Well, just let me back up a second. I think just so everybody's on the same page as us, basically mutations happen, but at a very low rate, right? Mutations happen over scales of hundreds of years, thousands of years. And we're talking about maybe 20 years, maybe since the Oregon blue tort was collected in the wild. Very likely, the genotype that we find in the frag in my tank is pretty much identical to the original colony that was collected from the wild. Yeah. And so, I mean, maybe we find different people having gotten different pieces by chance from that original colony in the wild, and they show up as being the same. But that seems to be kind of unlikely too. So I think that given the time scale we're talking about, and given what Eli is talking about, even if a mutation happens, the chance that it's going to be found in the entire coral, is pretty low. And so we think that we ought to be able to find some pretty recognizable genotypes that should be, pretty true to the original lineage.
So I guess, I'm trying to wrap my head around the process, like trying to poke holes in it to see, okay, how will this work long term? So, I mean, it seems to me there is an assumption that there is some mutation, though, that has occurred through aquaculture. Or there is some detectable difference of this strain being an aquaculture strain versus a wild colony. Because then you could differentiate, yes, it has been in captivity for 20 years. Am I understanding that correctly?
I don't see any reason to think that there'd be a variation driven by aquaculture. Just random mutation over time. The useful variation that we're talking about here is variation between different if you step back and think about it in kind of a human population sense, it's the genetic variation among different human families. Like here's a family that they all have red hair, and here's a family that they all have blue eyes. That kind of variation, and that variation has accumulated over millions and millions of years, right? That individual level variation in the population. That's the kind of thing that we're going to be looking at. That's the useful variation that we're going to be looking at in these gene sequences to see, is this individual or that individual. Now with corals, we have this other complicating factor that we clone them, right? We break off a piece, and now we have two things that are genetically identical clones. And yeah, I think Andy's right to talk here about scales, time scales. My expectation is if you broke off those two pieces and grew them for 100 years, passing them from one hobbyist to another, they'd be genetically identical at the end of that time period.
Okay, so given that, do you think that there will be enough useful variation between an aquaculture piece that has been in captivity for 20 years versus if someone has a fresh import from wherever the Oregon tort originated, and it's the same genotype from the wild? I guess would you be able to distinguish the difference between a fresh import that happens to be the same genotype from that same collection region, same mariculture, or not? Or would it just be?
I don't think so, Salem.
Okay.
I don't think so. I don't think. I'm not sure. I guess I'm not somebody who's done tons of diving, and Eli has, but my sense is that these different color morphs are not that common, right? It's not like you're going to go to some place where Oregon tort was originally collected and see just all kinds of Oregon tort, that it originated from probably a single colony, and that colony may not be there anymore. I could be wrong. Eli, what do you think?
Yeah, I think that's right. But there may be a lot of blue. There may be a lot of blue torts out there, numbers wise. Even if it's rare, there could be a lot of them. But stepping back,
they're going to be genetically identical in the wild, Salem.
That's right. Just because it's blue doesn't mean we should expect it to be genetically identical. I think maybe what you're getting at here, Salem, is just how variable are these sequences that we're looking at, and is that variation specific to like a population or a family or an individual? And some of this is for sure unknown, right? We haven't gone in and done population level sequencing of this species with these markers yet. That's one reason that we're setting out to collect a bunch of specimens from the same species, so that we can basically line up all those words together, and we can find that, okay, the first letter is always the same across all of these words. That letter is not useful. But this second letter, wow, look, it's different in every single one. That's a highly variable one. And so that's what we'll look at are these super variable ones. To directly answer your question, boy, if we had more wild imports from the same location, we could address that question directly, right? We could ask what we're calling the genotype of the Oregon blue tort. Do we find those same alleles, those same variants, types of the gene in that location? Or is it, specific to this? But for now, we'll just look among different torts and ask, is this letter different among the different torts or not?
Yeah, I'm trying to just kind of like thought experiment, steelman, civil discourse conversation of what is the scope of applicability for this as a potential product in the market eventually. So, in my mind, I'm thinking not everything is wild collected now. There's a lot of mariculture that goes on, and a lot of those maricultured pieces are named in lineage. So going back to the torch example, right? A lot of the lineage torches that are now named likely came from some of the same mariculture farms, in which case they would theoretically be clones. So in my mind, it's like, okay, if we have someone that's came in just like that guy at the shop, and they have a Top Shelf Aquatics Holy Grail, and someone bought the original from Top Shelf Aquatics, but there's also a fresh import, but both originated from that same mariculture rack, that same colony. Based on what I'm hearing, I don't know if it can actually, if this would be able to prove lineage back to Top Shelf Aquatics, the first people to name it. But I think that for pieces that were originally wild collected that were not mariculture, this could be a very useful tool. So that's what I'm trying to think of, is the scope of usefulness for it.
Yeah. So take an example like the one you just gave. I think it's useful to think about, how confident can we be in our answers? So in one direction, we'll be very confident. In the other, there's some uncertainties. Okay. These data will allow us to say for sure, confidently, these two are not the same. Mm-hmm. And that'll be ironclad. These two are not the same. I don't care if they have the same name on them. They're not the same thing. On the other hand, if we genotype two specimens and we get the same genotype from them, we get exactly the same letters lining up from these two specimens, you can imagine several scenarios that could lead to that, right? Perhaps they're not really clones of each other. It's just they're closely enough related that they're the same at these markers. So, just to put human terms, like they're cousins. They were collected from the same location. They are related. They're cousins, but they're not genetically identical. We can't exclude that with these tests, right? So, yeah, again, if the answer is no, it's a definite no, right? These two are not the same. If the answer is yes, well, maybe that means it's the true thing, right? Broken off the same colony. Maybe it means it was something that happened in nature, right? A fragmentation event in nature led to two colonies. Many possibilities.
So just to jump into quickly, to answer your question, Salem. So it might be that, say, with the example of torches, it might be that, say, Top Shelf Aquatics and Worldwide Corals get torches from the same mariculture supplier, and their torches came from the same source maricultured colony. Then those are going to be genetically identical, and then the name given by one place is different than the name given by another place, but this testing will tell us they're the exact same coral.
Yeah.
In terms of lineage from a maricultured colony, or as Eli said, perhaps one that's so closely related that they're just not appreciably different.
I think a very good test case for this would be the purple monster. So you mean the original colony from the early 2000s, 90s, whenever it was brought in. And I know that Meckley, last time I talked to him, and I think Keith, Reef Bum Keith, both had, and I know Johnny Shen, Thirsty Reef out in LA, he's got a piece of it too. But there's a lot of stuff coming in from the Solomons lately. And I know that Jake went to the Solomons and reportedly found where they originally collected. And now those are supposedly being imported again. But a lot of people that see them say they're way different. At least morphologically, they seem to be distinct. So I think that'd be a very good test case, is the purple monster, to determine, are these from the same, because then you have one with existing lineage, it's been aquacultured, and you have ones that are claimed to be collected from the same wild site. So I think that's like the only example I can think of to where we have something like that in the supply chain that would allow us to ask that question, if it's the same, and also if it is the same, if there's any useful variation between the aquacultured one and the wild one. I think that might be a good one to get a hold of. But if anyone wants to cut a purple monster for science, that's another question. I don't know.
That was on our list. That may have been the first one we talked about for this project. So yeah, if we can get our hands on it, let's do it. I guess our approach, I don't know. I think it was at Reef Stock this year. We just decided, like, screw waiting on other people. Let's do this with what we can get our hands on ourselves. And while we're doing this with Oregon Tort, let's keep talking. Let's find, people with lineage samples to do the project more broadly. Walt Disney, of course, is another great one. And any other tenuous, really, because there are so many tenuous in the hobby. There's a couple of fancy millipora too, right? Anyway, there's named millipora certainly that sell.
Just to quickly go back to the purple monster example, this is what makes me think that the original Oregon Tort it may be very, very distinct in its environment, right? Because they've been looking for purple monster for how long? It's not that
Quite a
easy to
while.
find it, right?
Yeah.
Even though they've gone back to places where it was originally collected, and they haven't been able to find it until recently. And then also that trip that Jake went on. So it's not like there's purple monster all over the place, right? And I suspect it could be the same for the Oregon blue tort. It might have been that the original colony that was collected was not super common. Maybe there were a few. And if so, I think we'll be able to confidently identify that by lineage.
I think another very interesting question this could help answer is, when you were speaking, Eli, of a mutation would have to go through, like, that clump of cells go through an entire colony to then have a recognizable or testable difference when you actually do the test. Well, I'm thinking of the traditional grafting phenomena, right? So the Jason Fox flame, very widely, lineage piece a lot of people have. I hadn't really seen it prior to Jason Fox. And when I went up north of Fort Collins here, the guys at the shop Underwater Wonders did a video on them. They had a grafted version of the Jason Fox flame, and they propagated out the green grafting. And they bought it, I believe, from Jason Fox originally, and the grafting phenomena developed in their tank. So I think it'd be very interesting to take pieces like that where we have known lineage pieces that have been grafted on their own to determine a potential mechanism for what really is grafting. I think that could maybe help too. Yeah.
Yeah, I agree. Yeah, the markers that we're using for this project would allow us to answer the question, is it a chimera? That is it really two different organisms, two different corals that merged early in development and grew into one colony? This is known to happen, at least in some macros, both in nature and in lab experiments. So our test could answer definitively if they were different genotypes that had come together. Perhaps we get the results back and say, no, the genotypes are the same. At that point, I would want to explore a different marker to ask, is it a mutation in the GFP, the green fluorescent protein?
I really think it could be a GFP mutation in a lot of these. So obviously for wild imports that are grafted, the chimera mechanism or explanation makes a lot of sense to me. But when you have someone buy a lineage piece where the original colony and subsequent frags for many are not grafted, and then all of a sudden it spontaneously appears in a system, that says to me there's a mutation that has caused that. And it's not because it doesn't make sense. Like, how would it all of a sudden there was chimericism and it's finally expressed in one person's system? That says to me it's something environmental that occurred. So I would be interested to take known chimeras, like the Ghanis that Meckley had, for instance, and things like that, and compare them genetically to ones where there's been this spontaneous appearance of different colors, and get a large enough sample size to say, here's the different mechanisms for grafting. I don't know.
Yeah, it's interesting because it's really it's a question we're asking about scientific mechanisms. It's a very geeky question. It's the kind of thing you publish a paper on. But the people
speaking
who
of that, Eli,
never care
speaking
about
of
this.
that, remember at Reef Stock, we were accused of being nerds. Joe Caparata said, listen to these nerds talking about coral genotyping.
Fair,
so just
fair.
It is maybe a geeky or nerdy topic, but just to bring it back to why we think this is really cool, besides the fact that we're nerds, it's cool because you can, if we can get this to work, which I think we will be able to do, if you have a coral in your tank where you've lost track of the lineage, but you think it looks like a Walt Disney, all you got to do is just sample a polyp, send it to Eli, and he can tell you, yes, with 99 point whatever % certainty, that coral originated from the coral that Mike Bigger originally imported. So that's the big picture here.
I'd like it to basically replace the trust me bro method of lineage. And you wouldn't have to do it on like every frag. If you've got a nice Walt Disney colony and you want to sell frags from them and say it's the real Walt Disney, do the test once and just say, yeah, my colony is the real one. Here's a certificate.
I think it also can be really a shot in the arm for the taxonomy of corals, at least maybe getting some new classifications. So if you're someone that's an importer that also aquacultures, Meckley, Route 66, there's a lot of people out there that do that. I mean, if you really look at things, there seems to be a lot of undescribed species. So if you've got the GPS location of collection, then you can get the genetic data to say, here's where it lies phylogenetically. There's some room for maybe new taxonomizing there.
I see a graduate research project for you there, Salem.
Yeah, that's right.
Yeah, I've got plenty of those under on the ideas list, man. But yeah, no. What I'm interested in is the grafting thing, right? If we can determine it is a mutation, and that's called get a work for 10 years and develop a gene drive and have a coral farm and make everything grafted, and there you go, money generator.
Yeah, I mean, so if it's not a chimera, then it's maybe a mutation, right? Spontaneous development of a new color. I think the wilder suggestion that people have come up with is that it's somehow getting from one coral to another, and you could wave your hands about mechanism, but get into GFP sequencing the fluorescent gene itself, and we could address that question. As I've said in the past, I'm definitely in the mutation camp. I think that's where it's coming from. But
so let me just say that corals are able to pick up symbiodinium from the water.
Yes,
sure.
And incorporate them intracellularly, right?
Yeah.
So it's not totally crazy to imagine some kind of gene expression factor being picked up at the same time, but
that could be worthwhile.
I agree that mutation seems more likely.
If it's not a mutation, then doing the 18S, or if there's any more specific markers for symbiodinium and testing the populations of symbiodinium between the two, I think that could. I mean, because there's more than just green that appears. Like I talked to Kevin Berta from Top Shelf, and he's like, yeah, we've got purple appearing on like Montipora setosa and stuff, and we'll propagate that out, which I don't know of any gene that's fluorescent protein related to purple, but I also don't. I'm not that deep in the weeds.
All the coral colors really are driven by. We can generally call them GFP genes. They're named after the green fluorescent protein from a jellyfish, but really it's a family of genes. They all look very similar to each other at the DNA sequence level. And they're all different colors, all the different fluorescent colors, the yellows, the reds, the greens, these all are so-called GFP genes. The blue and purple pigments that we see in corals, those are also GFP genes, but they're broken. They aren't fluorescent anymore. Now they just absorb and reflect light. And so they just serve as pigments. But so basically all the cool colors that we like to look at in corals all are driven by this same family of genes. Every coral has something like 7 of those different genes in its genome. And that's where we get all the diversity of colors, right? Because you make one tweak in one of the genes, and that gives you one color. Make a tweak in a different gene, it gives you a different color. And so in that context, it's really easy to imagine that a mutation can come in and lead to a new color, right? You've got several different targets for it, and if you do anything to these targets, it changes their color, basically.
Yeah, I'm really interested in kind of, eventually when you have time on your plate, trying to reverse engineer the cooking process. So seeing, UVA, UVB exposure, infrared exposure, different spectral exposures. And this would be good with a qPCR machine, upregulate or downregulate any of those different GFP groups to create a recipe for if you want this coral to express these corals or colors, you need this spectrum, this par, and then you could just, I don't know. I think that because that's kind of a thing that a lot of coral growers do now is like in their head, like, okay, if I place this coral at this par, it'll morph this way versus this way. But for the grafting especially, if you can get purple to appear all of a sudden, there you go. There's the ticket to wherever you want to live.
Yeah, what I don't know with the mutation part is there any way to? You keep talking about like applications for it, right? Somehow make it happen more. I don't know. Is there any way to control that, to stimulate that in the corals?
All
But it'd
right,
be
I
nice
have to say,
if that's what's
UVA,
happening.
UVB definitely seems to do some
Eli's
things.
gonna, Salem's going to make make the Reef Builders studio radioactive or something.
That's right. Yeah.
Yeah.
Pour a little radioactive something
Hey,
in
I
there.
thought of something that probably would be good to discuss. So, Eli, your eDNA test gets DNA from the water. Sometimes you get DNA from corals.
Right.
Some users have noticed that it will identify corals they don't have in their tank. So maybe
Yeah.
you could speak to different gene markers and their utility for taxonomy.
Yeah.
But and how we're going to do it
That's
differently
right.
in this study.
That's right. So the marker we use for the tank DNA test is not designed to be good at classifying corals. It does a really crappy job at classifying corals and
Fish
fish
too, right?
Yeah. And I've added notes in the report about this, but I think because they're at the bottom of the section, maybe they don't get read. I'm going to update the report to just collapse all that down, right? Basically, let's take the fish, for example, because it's a really extreme case. With fish, the 18S marker looks the same for all fish. And so the report simply shows you the one that was first on the list. I think it might be alphabetical, but it doesn't matter. The point is they're all the same, right? That marker can't tell apart fish from each other. And so I think the better way for me to report the information in the tank DNA test is to simply sum them all up and say, this is how much fish DNA you had.
And just a second to clarify, when we say marker, we're talking about a different segment of DNA.
Yep,
So
that's right.
Essentially one gene versus another when we say marker.
So yeah, that piece, that marker that I'm using for the tank DNA test, it's useful. We use it because it works across this very wide range of organisms, right? We can identify algae and other protists, animals, plants, fungi, all of these things with that same piece of DNA. But it's not great for classifying corals. So what we're going to do for this project is go in with different markers that are good for classifying corals. By looking at several different markers, that will remove the ambiguity. Because imagine one of them might boil it down to a list of, well, it's one of these three species, but I can't tell you more than that. It's one of those three. And then you look at the second marker and you say, no, here it can only be one of these two species, and only one of them overlaps. That's your species. By looking at these different markers, different parts of the genome, we'll be able to do a much better job of resolving it down to a species level than the single marker, single region approach that's in the tank DNA test.
All right. So yeah, we've talked a little bit about this. Why don't we switch gears a little bit here? What's new with reefability, Andrew? I think we've only talked about this once on in the live stream. You had not even launched it yet on maybe the beta test. So why don't you give people a rundown for what it is, how it works, what's new about it, and why I should not think that Skynet's going to take over my house by using this product?
Well, Salem, Skynet might already be in control of your house, so I can't speak to that.
I do have a smart thermostat that worries me sometimes. That's
Yeah. So Reefability is a company that has developed an app. It's a phone app, or it can actually be used on Macintosh computers, be used on tablets, Android or iOS, and it's a reef tank management app, as well as a data analysis app. And it has a number of smart tools. So you can use it on the simplest level. There's a free version where you can use it to log your chemistry parameters and keep track of the equipment in your tank. A number of things that you can do with some of the traditional data logging apps that are out there. But then the thing that our app also includes is a chat bot that we call Reefer AI that you can ask questions about your tank, and it will look at the data that you've logged for your tank and it's been trained to only pull from the data on reef tanks and reef keeping. And
So
so,
is that like, sorry to interrupt you, like Reef Central
yeah.
Post? I mean, what's training in AI for people that don't know? I mean, how can I trust that these answers are accurate?
Well, first, let me say that one of the three people in this video stream wrote an article that some people interpreted as instructions on putting peanut butter in their reef aquariums. Um, so just because something is AI versus written by an actual person doesn't necessarily mean that you can trust one versus the other. No, I'm just kidding. That was an April Fool's joke that Salem did, but it was a really good one and it fooled some people. Yeah, so it's an important point. I think a lot of people have a very layperson understanding of AI, because what you see is how it's used mostly on the web and mostly poorly to these web spinner websites that spin up fake stories that start with a headline and pollute the internet. That's how a lot of people see AI. Or in some cases, they see fake articles being written about sporting events and things like that. That is a tiny bit of what AI is. AI is used for all the most important data analysis and research at my university. It's being used to develop new treatments for cancer. It's being developed to restore the Great Barrier Reef. It is, the cutting edge tool for solving all sorts of major problems in the world today. And we are able to use AI in this aquarium app in a way that avoids a lot of the errors that other people may have seen in their own experience with AI. For example, we've trained the AI to not use sources like Salem's April Fool's article. So you will not see any references to putting peanut butter in your reef tank. And
Did
so
you
when I
just
say,
filter out anything written by Salem? Was that the
well,
just a
we could add a Salem filter, yeah.
Yeah.
So basically, training the AI is something we did over 6 months, where we fed it all sorts of reef keeping information and learned where there was not good information about hobby things and where there was good information about hobby things. And we put strict boundaries around the information that it could use. We also trained it to say, I don't know, when it didn't have enough information, which is key, because AI will sometimes, if it hasn't been trained correctly, it aims to please. It wants to answer your question. So sometimes it will make things up, which is called hallucination by people that use AI. It'll come up with an answer that looks good and sounds good, but is totally not grounded in anything. So our AI will not do that. So basically, what it does is it brings all the data for your tank together, and it does an analysis and looks for insights, looks for patterns. There's a deep learning algorithm that looks for patterns among the data and the actions you take in your tank, and then will provide you with feedback on that. It also looks in the larger group of all the users and what they're doing to their tanks, and it looks for patterns across all of the users. So that's a cool thing. And once we have enough people using the app, we hope to learn some important things about reef tank husbandry. But you asked about new things. One thing that's pretty cool is we recently, I said there were smart tools that were part of the app. One thing the AI is really good at is image recognition, and it can pull information out of a PDF and convert that into data. And so, for example, you can take a picture of handwritten data logs for your tank, and you can scan it, and it'll digitize that and put it into your tank log. It also will read data from ICPs, from Eli's microbiome reports, from the eDNA reports, from the NDOC test from Triton, all of those things. All you have to do is upload a PDF, and then it will pull out the important information and then provide you with actions that make sense based on those data. So one example of a smart tool is if you're interested in dosing trace elements to your reef, Captivate Aquaculture, or Reef Blueprint is the name for the version of the products available in stores. It was developed by Chris Wood, who was originally involved with the founding of Brightwell and is a really smart chemist, and has been keeping reef tanks for 30 years or something. Has made this great line of products, includes all kinds of things, but this tool uses his trace elements. And so what you do is you get your phone or your iPad or whatever and upload an ICP, and the app will calculate what your dosing regimen should be for the next 30, 60, or whatever number of days based on the deficiencies illustrated in the ICP. And Chris makes his trace elements mixable, so you can make a single cocktail where they're all mixed up in RO water, and you can calculate your monthly dose, or you can do it for 2 months. You hook it up to a doser, dose the same amount every day, and that can address 18 different elements at the same time. So that is a tool that the app is made really easy to do. And so we're almost ready to release a second version of that, where you can incorporate the previous month's dose. And so what it'll do is it'll calculate, Chris's spreadsheet calculates what the normal monthly use of your trace elements is, and then also what you need for a correction based on your ICP. And so you can incorporate the last month's dose when you determine what the next month's dose should be. So just an example, one way that the app can make something like trace element dosing easier. Sorry, go ahead, Salem.
So you mentioned a free version, paid version. How much is the paid version? And then what do you get? What's the difference between free and paid? Like, what features are we missing if we're a free user?
Yep, good question. So the free version right now basically allows you to log all your chemistry data by hand. That is, not using AI tools, because the AI tools require that we pay for the service from a number of AI providers. So we have to charge a subscription for that. But the free version does incorporate some AI. It does incorporate the Reefer AI chatbot. So there's that, plus the logging of parameters manually, where you input your alkalinity and calcium or whatever you want by typing in the numbers. And you also have access to ask for a reef specialist. So we have a registry of people that do maintenance on aquariums all across the country. And you can look to see if there's somebody who signed up as a maintenance expert in your area. If you're having an issue with your tank, you want to have somebody come out and look at it. And then we're also introducing a marketplace so that people can sell and buy corals. So that's all on the free version. Then on the paid version, that gives you access to all the rest of the AI tools. So being able to upload a PDF of whatever data type, the Captivate trace elements tool. We have a BioLoad tool where you can keep track of all the animals in your tank, and then it will assess whether a new addition is likely to be problematic or not. There's a pest identification tool where you can take a picture of a pest and write a sentence description of it, and it will give you a likely ID and tell you what you ought to do about it. So there's a lot more tools and that use the AI that we're expanding those every time there's an update. Oh, and I forgot to say how much it is. It's currently $39.99 for a year, and you can pay monthly. I can't remember what it is per month. I think it's like maybe $12 a month or something like that. if you choose to pay
Okay.
monthly.
So now that we've heard what's new with aquabiomics and reefability, I think I'll transition to some general questions for both of you that I've seen some people ask. So to kind of give you all an opportunity to answer questions that I see people ask about both services. The first I'll ask, which I think applies to both of your companies, is kind of a question of the database sample size and what is an ideal tank. So one point of pushback I've seen for aquabiomics is how do we know this is the ideal or healthy microbiome of a tank? Because, or how do you determine that a tank is a successful one based on the input variables? And I think the same applies for your service, Andrew, based on the fact that the AI model can draw from the entire database of systems. So I think that's the initial question. How do we know what is good? And how are we building the average? Is it an average? Is it the ideal? What is the actual representation of the microbiome? And where is the AI pulling advice from, so to speak?
Right. Yeah, it's a question that comes up a lot for sure. How do we know what's the ideal, what's the best possible microbial community for a reef tank? We don't. Science doesn't. We don't. I'm actually Ryan from BRS said something good along on this question at one point about our microbiome data. Basically, I can't tell you what the ideal conditions are to grow corals in, but I can tell you here are the conditions that work. These are conditions you can achieve in a reef tank in a terrestrial living room somewhere, right? It's probably quite different from nature. It may very well not be the ideal. It looks different than the microbial community on a natural coral reef, right? So just for that reason, I think we could suspect it's not ideal. But it's a very large database, thousands and thousands of tanks, and almost all of these tanks have healthy communities of corals and fish and invertebrates in them, right? So this is a database of what the microbial community looks like when it works. And we exclude from that database in terms of coming up with, what is the typical tank that you want to compare your tank to. We exclude from the database things that we know are problems. We have, people send us experimental tanks, and okay, we're going to exclude those from the database, aquaculture systems, things like that. But the fact is, as the database has grown, it has mattered less and less what I exclude from the list. I keep noticing that when I add stuff to my exclude list, it doesn't actually change the picture anymore because there's so many normal ones in the database now that even having a couple abnormal ones doesn't change the picture very much. So yeah, what we're calling the typical database truly is the average of this very large collection of tanks that work, that succeed in keeping corals and fish alive.
Okay.
I think,
Andrew.
Eli, just a quick jump in there. I think that one of the things that you were talking about with a user was potentially differentiating what an average tank that, say, runs UV or an average tank that runs ozone, which might look a little different, might look like, right? So that's one way you've thought about maybe expanding that a little bit, right?
Yeah, I've been playing with the code for those reports this week. It's not, my new version is not ready to go, but it's in the works. At some point in the future, the report is going to have the ability to compare your tank not just to the typical tank, what we've discussed here, but also to the typical tank that uses UV, or maybe some famous and interesting tanks, tanks from coral aquaculture facilities, tanks from coral research facilities. I'm still thinking about what other tanks to include, but I need to build this mechanism first, the mechanism where you can compare your tank with multiple different reference points. And certainly the first one we've talked about is compare it to other tanks that also use UV, because that has such a major impact on the microbial community. Yeah,
that would be, I think, some welcome expansion, because a lot of people, I think, they've not understood it as this is the average collection of tanks that are not crashing, that are not having abhorrent issues. And I think people have mistook it as these are ideal tanks. And I think it would be interesting to take a look at truly ideal tanks. So top shelf, the top 10, 20 SPS farmers in the country, and then filter those down, the ozone, the UV, yes, no, and then say, here's the top 10 acro farmers in quotations in the country, what their microbiomes look like. Yeah. Things like that, I think, an average of the Great Barrier Reef with publicly available stuff and published studies. I think those could be very good reference points for people.
Yep. Yeah, I absolutely agree. I mean, there are going to be differences, but there's also going to be some real similarities here. So, this pattern of what the typical tank looks like, this pattern emerged very early in my collection of data. And that's because so many good tanks ended up looking like this pattern. Andy's is a very nice example. Andy has a beautiful tank. When I run a new batch of samples, I often have one of Andy's samples in there. And as I'm looking through the reports, the first report I pull up, oh man, this one looks weird, right? They've got no Pelagibacteriaceae or something, right? The second one, oh man, this has another pathogen in it. Oh man, is this whole batch weird? Right? But then I get to Andy's tank, and Andy's tank looks just like the typical profile, unless it's an unusual month for Andy's tank. My point is to say that if you were to compare your tank results with a good tank, it probably wouldn't be that different than comparing them with this typical tank result, because a good tank basically looks a lot like this typical tank result. But there will be some differences. Just on this note of Pelagibacteriaceae, the kind of big obvious difference. If you compared your sample with Andy's, you would see that his sample has a very big pink band, right? A high population of Pelagibacteriaceae, more than the typical tank. So comparing your tank with Andy's tank would lead you to conclude that your tank is more deficient in this group than the comparison against the typical. Do you see what I mean? So, I mean, it is going to change. This ability to compare it with multiple different reference points is going to change the conclusions you come to. But I think
the first choice should be compare to Andy's tank.
You're the gold standard.
A good tank will look generally like this typical tank profile, aside from things like UV and some of these other issues.
So then, Andrew, how does Reefability, the AI, is it learning from the database writ large? Do you have restrictions on tanks that are, exclusions of tanks there? How are we not getting some incorrect data feeding off of the larger database?
So when it comes to microbiomes, that's a really good question, Salem. So you put your finger on what took me a long time this summer to figure out. So there's not a lot of information out there about aquarium microbiomes, right? You ask AI to go look on the web for information about that. It's going to pull up something on, human gut health or something, right? And that's not really, it could be relevant, but it's usually not relevant, right? So we had to train the AI to work on a very limited number of resources. And so those resources are based on this average, successful, mature community that Eli has collated over 5 years, right, of thousands of tanks. And so it's based on that. That is, with caveats that if you have UV or if you're using ozone, you're not going to have certain families, right? And so it's starting with that information. And over time, it should learn what other possible successful communities look like, right? And, for example, it looks for cause and effect, right? So it looks at the data that are being entered, and then it looks at what's being reported in the tank at the same time. So if a user is reporting lots of problems with cyanobacteria, it's going to look at the community. It's going to look at your water chemistry. It's going to look at your parameters. and it's going to provide recommendations about how to make a correction. It might be that your NDOC level is like 3 times the maximum, right? Which has actually been something that I've seen to be correlated with problems with cyanobacteria in my own system, is a higher NDOC level, right?
Yep.
So if it starts to find information like that among the larger database, then it's going to make that recommendation whenever it sees somebody with a higher NDOC level, that, hey, look out for cyanobacteria issues potentially. There may be other things that contribute to cyanobacteria. As more and more users use the app, as we have more and more data in the app, as we have more and more data from Eli, right? We're gonna have more possible solutions, more possible stable communities. We're gonna know more. So we had to start out kind of knowing what we know from Eli's data as they are, but we hope to go further along in our learning once we have a lot more data.
And this sounds magical, Andrew. You're describing a system that can diagnose things we don't even understand yet in the industry. Is that what you're talking about here? Is maybe 5-10 years down the line, it turns out that dinos are caused by high molybdenum levels every single time, and we don't even know yet, and it can just tell us that, and it's the cutting edge. It's what it sounds like.
Currently, Salem, it'll tell you that if your nitrate is going too low, that you're at risk for dinoflagellates. But yeah, I mean, as we have more ICP data, as we have more microbiome data, hopefully, we'll learn more. I think that there are multiple avenues that lead towards dinoflagellates, right? There's multiple ways you can end up there. But one of them that clearly happens to me, and it just happened this last week, is when nitrate gets low and phosphate creeps up, there's a certain dinoflagellate that always pops up in my tank. And it usually takes about a week for me to correct it. I just have to dose some nitrate or ammonium, and it goes away. And so we're going to hopefully learn more things like that as we have more data from more systems. Yeah.
That's what excites me.
That is the potential I'm excited about for this this project. And I find it really exciting because I've been saying the same thing for 5 years. Like, as the database gets big, we're going to learn more from it. But what I keep not admitting or not saying out loud is really there's a step 2. The database has to get big, and then I personally have to sit down and do some stuff. I have to write code, and it won't work, and I'll have to rewrite it. It takes human time to learn from the database. So I'm really excited about the potential to take that human time element out of it and have the robots do the learning for us, and to help facilitate this growth of the database. I'm including a link in the Aquabiomics reports where you can just click and upload it directly to Reefability, to facilitate that feeding data into the robot brain that'll teach us.
So does that happen even if you do not have a paid subscription for Reefability?
Good question. How's that work, Andy? I mean,
I don't know if we've worked that out yet.
We're still testing the link, but we're
still working that out. So to be continued.
Okay. So Andrew, I know you have to leave in about 5-10 minutes here. Anything else you want to talk about before you head out? Any other common questions, news, things you want to speak on?
You can learn about new things by either following me on Instagram. I'm on Instagram as Lex Inverts, LEXINVERTS, or Reefability. We're we both are on Instagram. I'll be posting stuff, and Reefability will be posting stuff. So any new releases and stuff like that, new data as we learn new things, I try to post that stuff to Instagram. So yeah, check us out there.
Awesome. I think we should get down to brass tacks here, which is I've heard some reports, Eli, that you're trying to destroy companies. There's been some talk online and forums and such that Aquabiomics is the death knell of the industry, so to speak. And it's certainly something that's not very good and is really making some companies upset and changing some consumer behavior. Do you have anything to say in regards to those messages?
Some harsh words. Thinking about bacterial products here. Is that the topic we're on?
That's been the common, or not the common, but that's
big
been
bacteria.
Yeah, bacterial products.
Yeah, so I mean, I have nothing against these companies, and I'm not taking any active steps. What I simply do is when people send me a sample, I tell them what's in the sample. Right? I'm not. I don't have any campaign to prove or disprove any theory about bacteria in a bottle. I think there are some misconceptions about the use of bottled bacterial products in our hobby. And I think that some of the data that have emerged from our tests help to disprove some of those misconceptions. But it's not a part of any campaign of mine or any effort of my company. You know, it's simply people send us samples and they learn from the samples that they send us. I'll tell you one of the things, you know, and this is a criticism I will make of the industry. I feel safe making this statement. One of the things that has emerged from our testing, and interestingly, somebody on Humblefish recently posted their own independent testing. They work in a DNA testing lab. They did their own testing of some products. And the same conclusion emerges across manufacturers and now across testers, myself versus this other guy on Humblefish. And the conclusion is this: quality control may be non-existent in this industry. What is in your bottle is fairly variable from bottle to bottle, batch to batch, manufacturer to manufacturer. It's been troubling how much of that we've seen. Buy 3 bottles of it, and they're fairly different. Yeah, you know, there's other topics that could be discussed in this. That is, why aren't you guys putting labels on your products that you're selling? But I think that the variability, the consistency in the product, maybe that's a safe area where we as a hobby and manufacturers can agree. At least we should be able to agree that every bottle you buy of this product should be the same as every other bottle of the same brand.
Can I say 2 quick things before I have to go?
Yeah.
Number one, don't use bottled bacteria products to increase the diversity in your tank.
Yeah.
Eli sells sand and rubble that have between 600 and 1,000 different bacterial variants in them. Most of the bacterial products have 1, 2, 3. Okay.
Yeah.
So use something like live sand or reef rubble to increase the diversity in your tank, if that's what you want to do. It doesn't make sense to use bacterial products for that. Number 2, if I was a company who produced a bacterial product and I did not want to be surprised by somebody's results after sending them to Eli, I would consider sending my product in to be tested on a regular basis myself. I think that makes a lot of sense. I know that Hydrospace sends all their stuff to Eli all the time, and it's no mystery what's in those bottles. So
that's right. And boy,
it makes a lot of sense for manufacturers to do that.
Absolutely right. Yeah. And you know, we have a couple of manufacturers. Ken is one of them, who've used our service regularly. I don't know any manufacturer that's doing sort of batch level testing, you know, every batch every time. But we're open to it for sure, and I bet we can beat the price of any other lab that, you know, manufacturers wanted to go to for this stuff. My own position is I think this stuff should be tested on a batch level at some level of testing. You know, does that mean DNA sequencing? I don't know. We can talk about details, but I think every batch should be tested on a batch level. And I think the active ingredients of these products should be disclosed. If you want to have your own special strain of a species, okay, you know, fine. Tell them, well, it's my house strain, but tell people what species is in the bottle. I don't know. That's my position based on the fact that that's how it works in like every other industry, right? I mean, I can buy nonsense homeopathic remedies and they're at least going to tell me what 1,000-fold dilution of flower juice is in this bottle, you know. But here we don't even get that information.
Yeah, it's a
Well
cutthroat
said.
industry.
Okay, guys, I got to go.
All right, thanks for
Thanks a lot
coming
for
on.
having me on, Salem. Take
Good talking
care.
to you.
Yep, we'll catch you next time. Thank you. Yeah, so I think I view this as the new ICP. So before Triton came on the scene, there was a lack of quality control, or at least higher variability among many of the supplements and salt products. And I think that at least the people that I've talked to, I've spoken to a lot of the manufacturers on the dry goods side that produce these bacterial products. And I think the main distinction that I've kind of caught on to is that the lack of quality control certainly comes from the industry-specific manufacturers. So people that are growing their own bacteria. A lot of people just order bacteria, strains that they see as beneficial for a system from a third party, from a biotech company that does grow them with some quality control. And at least from the data that I've seen, there has been more consistency in the companies that I know order from third-party growers. A lot of the inconsistency I've seen is people that are doing this on their own for the industry. And I think that that's where some prodding or some change would be very welcome, because there's not only the risk for contamination, but there's obviously that variability to where the product may not perform as advertised for every single bottle. The dosage may change if the concentration's different, or just a random, not even like a pathogen or a bad contamination, but benign contamination grows and your bacteria of interest decreases in population. So yeah, I think that it would be welcome and beneficial long-term for there to be that level of quality control. Now, will that happen organically, naturally? Who knows? I think that as people are more aware of these things, that there will be a natural shift. And I don't think that anyone that's really talking about this is trying to attack these companies. It's more so an urging to say, this is something that ought to be there. There should be this level of QC in manufacturing because, there is liability. This is not, there's the liability of crashing your tank if you have a bad salt batch, but if you've got a bacterial product you're growing up, there could be human pathogens, fish pathogens, coral pathogens. There's some real civil liability there that I think these companies ought to be concerned about, to be honest.
Yeah, I really think they should be concerned about it. You know, it's not just my opinion that some of these bottles had human pathogens in them. If you took them to other labs, you would also have found a human pathogen in them. So, I don't have any kind of regulatory authority. I'm not coming in to get anybody in trouble, but if you got a bad enough infection and wanted to sue a company, that company could at least get some headaches from it, right? I'm not going to cause them any headaches, but somebody could.
And that's not my goal either.
Yeah, I think it's in their best interest too to do more testing. This is, they're live products, right? It's a live bacterial culture. That's what they're claiming that they're sending you. Those things are really hard to keep pure and to keep stable. So it may be that the industry, in terms of distribution, needs to think about storing them differently. Maybe the shelf lives need to be handled differently. Maybe there are manufacturing processes that need to be adjusted. And I don't have any opinions about any of that stuff, but I do think in the hobby we should expect that every bottle is the same.
Speaking like not even just in terms of consistency, but my concern is really human health. So there have been some of the products, at least the batches that were tested, they did have some potential human pathogens. And that's not to say that every bottle or product that company sells has that, but it is to say that if that's in there, that shows that there's a lack of quality control and likely some contamination issues.
Yes.
And I think it only takes one person that's immunocompromised to buy that product that could, God forbid, die from it. And there could have already been people or instances where there has been some complications, but the necessary data proving that link was not there yet. So I think it's a ticking time bomb. And any company listening to this, this is not to attack you. This is not to make you go out of business.
Yeah,
It's
that's
to
right.
it's to make this better for everyone, and it's probably a necessary step that ought to happen.
Yep. Yeah, maybe an inevitable step, even if we hadn't come along. You know, somebody else would have come along with testing. Yeah, your comparison to ICP and salt mixes is a good one, right? And nobody after that, you know, after we realized how much variation there was in salt mixes, nobody came out and said, "Oh, well, that's okay. The companies should be able to keep it secret." You know, everybody said, "Hey, we need to fix this." You know, and now often you get sort of a certificate of analysis with your salt mix, right? That says, "Yeah, this is what's in it."
It's very commonplace for many manufacturers to have their own machines at this point and do it in-house even. I mean, I know several of the large dry goods manufacturers have MS and OES on deck and are doing it. And many of these companies have labs as well, to where at the very least they could do, you know, Vibrio plates and testing like that. And maybe it is done, but it's certainly not advertised that it is.
Right.
So,
right. Yeah.
Yeah, I think you know, going from this conversation, another question I will ask that I have seen some people ask is, how do we know that you're not changing the data? How do we know that you really don't have bias? So I think maybe a general overview of what you actually do with the sample, and maybe just a very simple, basic explanation of how this machine works, how DNA is sequenced to where the results aren't really variable. There's not false positives, false negatives, so to speak, and it's a very consistent or trustable process.
Right. Yeah, I mean, you know, I guess at some level any service provider could falsify data, right? You have to have some level of trust. But I'll tell you how I handle the samples, and how I handle the samples is in a very blind manner. I don't even know whose sample I'm handling until I get to the very end of the process and I get the report, and I open up the report and I look up on my spreadsheet, who does that belong to? You know, throughout the whole process, I have it labeled with just a code number. Would have no way of even knowing what sample to modify if I wanted to modify one. I mean, let's talk about the method itself. I've always said that the reason for the turnaround time is because we have to process things in a batch, right? I can't just, you know, when you personally send me a sample, I don't go into the lab and work on just your sample. I go into the lab and work on a batch of 24 to 96 samples at a time. Each one of those samples is just in a tube with a number on it, right? It doesn't say Salem sample here, be careful with this one, but this other jerk, you know, do whatever you want with his. You know, so I process these samples in a batch. I take your samples and I extract DNA from it. What that means is I grind it up and I get rid of everything that's not DNA until I'm left with just the DNA in a tube. I put that through a whole bunch of steps in the lab that I won't bore you with the details, but it's all about taking that DNA and making it ready to put onto a DNA sequencing machine. So I do those steps individually to each sample, but I do them all side by side. Right? I have all these tubes laid out side by side. And I just do the same thing to every one of those tubes. Once I get the samples ready for sequencing, I've modified them in a way that now they can go onto a sequencing machine. Then I send it off to the people who own the sequencing machine. And then I do it that way because it costs $500,000. I can't afford the machine. Right. Yeah. Actually, that's how most researchers do it. So they put it on the machine and they send me back these files with just thousands and thousands of DNA sequences in each file. Literally just letters, ACGT, thousands of times in a computer file. And again, all this is still anonymous through the process, right? It's just a coded, you know, this is sample number 3. And then I push it through some computer programs. These are programs I've written. Some of them use other software that other people has written. We call it a pipeline, basically a series of steps. You know, the data is going to come in at this end of the pipeline. It's going to go through a series of steps. And when it gets to the end, it's this report, this HTML file that I put online. And really, that's the point at which I look at it and say, okay, did it work? Do I need to rerun the sample? That kind of thing. And that's the point at which I discover, oh yeah, this is Salem's sample. Huh, I wonder how his tank is looking. It's just at that end of the process. Yeah, I don't know. So that's a 10,000-foot view of what it looks like for me to take your sample and push it into the report. Happy to talk about details.
Yeah, I guess maybe is there any potential error to where when you have the samples side by side, could you get my sample mixed up with Ken's sample to where the data might be a little bit different?
Right. Yeah. I mean, so cross-contamination is something we think about a lot in molecular biology because, yeah, you're handling multiple tubes, right? So, I mean, I'll say a few things here that, forgive me, they may seem obvious, but, we do things, we take some precautions to prevent that. So as I'm adding liquid into every tube, to do the things in the lab that I talked about, the way we do it is by adding a microscopic amount of liquid into the tube. It's a chemical that makes something happen, right? And when I do that, well, I use a new tip for each one of the samples, right? It's a disposable plastic tip. We burn through a terrible amount of disposable plastic material in this process. There's just no way around it. And we do it exactly to prevent cross-contamination, right? A new tip for every single chemical that goes into every single tube. I burn through thousands of them. Not only that, those tips have inside them, if you got the tip out and looked at it, there's a little piece of kind of a filter, cellulose filter in there. That's to prevent any droplets, tiny aerosol droplets, from floating up into the pipette, the thing you're holding, and then getting pushed back down into the next sample. So not only are we, changing the tips each time, but actually the tip prevents little particles from getting transferred. On top of that, I only open one of the tube lids. Each tube has a lid with a hinge on it, and I only open the lid for one of them at a time. All the other ones are closed. Lots of little things like that we do in the lab to really minimize the chance of cross-contamination. And then I also look for it at the end. So when I get to the end of the process, I've had these tubes oriented in a certain way, rows and columns throughout the process. And when I get to the end of the process, I actually have it still mapped out. It's 96 little holes in a plate. And I ask, okay, if I found uranema, did I find uranema like all the way down that column or all the way across that row? I mean, that would indicate, contamination had happened. And we don't see that because of the precautions we're taking. But that's a way of looking for it, looking to see, well, the two samples that were next to each other, do they have the same thing in them or not? Yeah.
So I think that kind of can answer a lot of the potential concerns about, you doing things in the lab, if that can alter the results. I think another question that I see is, people are very familiar with the existing types of analytical equipment that are used in the hobby. So an ICP, MS, or OES, or a fluoride analyzer, or total carbon analyzer, whatever. All of these things need to be calibrated. There's algorithms that are on the software side that can filter out results to where they're optimal. And obviously those things can have false positives or false negatives based on how they're calibrated. How is a DNA sequencer a little bit more reliable in those traditional machines? I mean, how do I know that if you've extracted DNA and there's no cross-contamination, that the DNA it reads is truly what's in there?
Yeah. So let's compare DNA sequencing with a basic chemical test for nutrients in your water. So we call these tests colorimetric because the color changes as you add more of the nutrient, right? You'll get more of a green color if there's a higher ammonia level, right? And less green or more yellow if it's a lower ammonia level. So the color changes. So what's really happening there is some complex chemistry is going on, and the eyes or the machine, if you're using a machine to read it, is just saying, well, there's this much green here. And it makes the assumption that much green means that much ammonia. But really, there's lots of other things that could be making it green, right? There's a lot of ways to make a liquid green. There could be other chemicals that lead to the same chemical reaction. There could be other just green contaminants in the liquid would also make it green. And you're simply interpreting everything that's green is ammonia. The same thing is true with ICP, except it's a peak on a chromatogram instead of a color. You're saying everything that is that peak is calcium, and everything that's that other peak is magnesium, right? DNA sequencing is very different than that. It's not just looking at the amount of something and saying, oh, you have a lot of green, therefore you have uranema. DNA sequencing is really taking a linear molecule. So it's a molecule that's a chain, like beads on a string, right? And it's reading each one of those beads in a row. Now it's reading them through a chemical process, and there are errors that can happen. There are contaminants, right? There are things that can go wrong. But all of those things that can go wrong would cause us to basically throw away that piece of data. So I get thousands of DNA sequences back. If there are sequencing errors in those DNA sequences, I automatically throw them away. My programs say, no, that's a bad one. I'm going to throw that away. I'm not even going to look at it. We only look at the ones that are very high quality sequences, and we line them up. So now I'm thinking about this sequence. It's like a word that's 300 letters long. There aren't a lot of words 300 letters long, right? If you were to find two strings of letters that were exactly the same for 300 letters in a row, you'd probably be confident saying, yeah, that's really the same word. And that's exactly what we're looking at here. It's not simply more of something like, a higher fluorescence signal or a higher green color. It's finding a discrete thing, a discrete word, exactly this string of 300 letters, and saying, well, that can only come from uranema because nobody else has that particular string of. So it's a discrete data type, not a quantitative data type. It's a very different kinds of errors could happen.
So I think for people listening, what this means is if you take a sample of your tank or a bottle of bacteria and you send it into Eli, there's a very low probability that what you see on his report is not what's in that bottle. He's got lots of redundancy, lots of processes that would eliminate things that would not really shake out in terms of the data working. So this is not the traditional type of reliability you have with a normal little titration test kit to where you have to determine the color change based on a little color card. This is a very precise, very exact, and it's very cutting edge, expensive technology that the best labs in the world, whether they be the Smithsonian or Harvard or any lab you name, they're using the same technology to do things with human medicine. Pfizer, vaccine development, very expensive processes. And Eli has taken this and gave it to the aquarium industry, which is awesome. That's why from the beginning I've been very excited about this, because coming from a molecular biology background in college, this was technology that we used to do any of the research projects. And then all of a sudden Eli says, I'm choosing to leave academia and bring in the same process here as a product. So I think as a final kind of landing point, if people have not heard of you before, if they're interested in getting tests for their tanks or getting the live rock, live rubble, where can they go? Where can they find you? Where can they see updates? Where do they buy and shop Aquabiomics?
Yeah. So right now it's all directly through our website, aquabiomics.com. We do have a little bit of social media presence, but I don't have the time for it, or the natural inclination. Let's be honest. So we do have a Facebook account. You can message us there, but the website and email is always the best way to get in touch. Yeah.
All right. Anything else you want to talk about today, Eli?
No, I guess I'll leave you with kind of the news and the timeline of where we're going next. So I've just mentioned that we're moving to three batches a month. So that is every 10 days, we're going to do another batch of data. So much faster turnaround time. But we have some other exciting developments that are coming really right on the heels of this. Right after that, we're going to launch what I'm right now calling Microbiome 2.0. So this is a fundamental improvement to the microbiome test, very specific to the Vibrio pathogens. So right now, in the current version of the microbiome test, we can't tell all of them apart. So we can identify some Vibrio pathogens, but there's others where all we can tell the client is you have Vibrio of some kind, and it's a really high level or a low level. That's where we are today, but I have Microbiome 2.0 completely working. We've done it on a bunch of samples, and it's working on all samples now. This will give conclusive species level ID for all the Vibrio pathogens. So that'll be rolling out within a month or two after that. And along with all of this, I've been working on a lot of updates for the reports themselves. So expect within the next 6 months to see some major updates to the reports too. Yeah, so that's my timeline in the near future.
Yeah, the Vibrio thing, that's very exciting because for people who don't know, the kind of family Vibrio, so to speak, a lot of those are putative pathogens. A lot of them can cause problems, but inside of a coral, there's some members of that family. There's some Vibrio species or strains which are very good that actually help with the coral's immune system and nutrient cycling, and they don't have any pathogenic potential. So even if you have really high levels of Vibrio in your tank, that could theoretically not always be a problem. And having the species level identification can allow you a more accurate diagnosis to say, okay, I've got a lot of bad guys or potential bad guys that are in my system versus I somehow have only the good Vibrio and they're really there. And you might not want to do something like a Cipro or oxalinic acid treatment in that scenario. So that's very welcome news. That's great. I did not know about that. That's cool.
Yeah, and there's real variation there in the tanks we've tested so far. It's not like the old legends, how they used to say, well, everybody has ick in their tank, right? And it's just about how you manage it. Our testing has certainly showed that's not true. And it's not true for Vibrio pathogens either. Not everybody has at least measurable levels of Vibrio pathogens in their tank. Very often when you have Vibrio, it's one of the harmless kinds. But boy, it'd be nice to know for sure if you have one of the nasty pathogens, and the test is ready. So we'll roll that out in the next couple of months.
Well, cool. I appreciate you coming on the podcast today. To all the listeners, thanks for listening to this episode. Be sure to like and subscribe. And this episode is brought to you by Cali Kid Corals, the ultimate coral experience. And we'll see you on the next episode. Be there, be square.